Okay, so ladies and gentlemen, you are all welcome. So in today's lecture, we are going to look at the omics approach. And my name is Mukhtar Ali Ikara from the Department of Biochemistry. Introduction to omics. Omics is refers to a set of biological disciplines that aim to analyze and understand complex biological systems. So when we say complex biological system, it can range from cell, the tissue, organ, and the entire organism itself. So an omics approach is an approach that's actually trying to analyze and understand the complex biological system using molecules and also some biological mechanisms. So this omics approach, it aids to analyze molecules in cells, tissues, organ, and the entire organisms. So that is why when we say omics approach, it's an approach that tries to understand the complex biomolecules that are found in living organisms or a component of the living organisms, like cells. Like, for example, you can actually study an omics approach or an omics of bacteria, omics of fungi, omics of algae. That is how you can study a complex biomolecules, analyzing them in bacteria. So therefore, an omics approach is an approach that tries to analyze molecules in a cells, tissue, organs, and the entire organisms. And these disciplines or this approach include genomics, it includes proteomics and metabolomics. It also includes transcriptomics, epigenomics. These are the omics disciplines. So under the omics, there are sub-disciplines under omics, like I can say like sub-branches of omics. And these sub-branches of omics include genomics, proteomics, metabolomics, transcriptomics, and epigenomics. So this is actually the approach. So we started with actually phenomics, metabolomics, proteomics, transcriptomics, epigenomics, and then genomics. But the age and environment are the two major factors that also affect this omics approach. Because the types of the genomics data that I will have as someone living in Nigeria might not be exactly the same with someone that is living in Europe, because the environment and their environment and our own environment in Nigeria are two different things. Age also. So these are the two major factors that affects the omics approach of an individual. It depends on his environment, or it depends on the environment of the individual as well as its age. Like, for example, I as a human, you cannot actually assume my omics data as of my age now and in the next 20 years. It might not be the same, or in less than 20 years of my age. So omics approach, we are just talking about molecules that are found in the biological systems. So how these molecules in biological system integrate themselves to produce a functional living organisms. So that is why the omics approach always started from genomics. So it started from genomics. When we say genomics, it means the entire study of the DNA in a particular living organism. And of course, with the help of epigenomics methods, the genomics, which is actually the genome of a particular living organism, let's say DNA, then it will now convert it to transcriptomics. So the transcriptomics is the study of the RNA, then proteomics, which is the protein, then metabolomics, and then phenomics. So phenomics is the physical appearance, that is the phenotype. So, ladies and gentlemen, before I go deep, we need to understand some very important information here. From the genomics, so genomics, as I said, the genomics, it means DNA. It means DNA. So, of course, from the DNA, we'll go back with the help of epigenomics methods to be converted into RNA, which is the transcriptomics approach. So from the DNA, it will now go back from the DNA, it will now go back to RNA. It will now go back to RNA. RNA, that is ribonucleic acid with the aid of epigenomics method. And then, of course, from the RNA, then it will be converted into protein. So the protein is the study, or the proteomics is the study of the entire proteins in a particular cell or in a particular living organism. And then the next one is metabolomics. That is the study of the entire molecules. It can be nucleic acid, it can be carbohydrate, it can be proteins. So the study of all these metabolites is known as metabolomics. And then phenomics is the study of the physical appearance or the phenotype of a particular living organism. So what I wanted us to actually say here, ladies and gentlemen, what actually makes us to have the appearance, the characters, and everything that we have is actually coming up from this omics approach. So because DNA is a language of life. So we look the way we are intelligent, the color of our skin we appear as the way we are because of this omics approach. So starting from DNA, RNA, proteins, then the metabolomics, which include proteins, carbohydrates, lipids, and nucleic acids, with some also other biomolecules or other metabolites that are found in living organisms. And then these metabolites will now actually translate into our own phenotype that actually have. So the omics approach, starting from genomics to phenomics, are actually affected by our age and our environment. So that is why this organization of this omics approach, you can look at it from the organelle level. That is from, let's say, like mitochondria, nucleus. Like, for example, starting with the genomics approach, like DNA, which organelles do we have? It's a cell. Like in human, we have this DNA in mitochondria and the nucleus. Then we can look at this omics approach at cellular level. We can also look at it at an organ level, and we can also look at the omics approach at the organismal level. So then let's start with the genomics. So genomics is the study of the complete set of DNA within an organism, including its genes. Like, for example, in humans, we have this DNA, and it's found in nucleus and the mitochondria. So the genomic DNA is found in the nucleus, and then we have also the mitochondrial DNA, although they serve different function. And in plant, apart from the nucleus and apart from the nucleus and mitochondria, plant also have DNA in their chloroplast, and they are not the same. They have different functions. So the study of the complete set of DNA within an organism, including its genes, is what we call actually genomics. Like, for example, in humans, the human genome contains about 3 million base pair. 3 billion base pair, not million. 3 billion base pair. It is actually a project that is conducted in 5 different countries, which take them 13 years. It started from 1990 and end of in 2003. So the Human Genome Project is a project that actually carried or that was conducted to sequence or to have the complete sequence of the DNA. That is the latest that form the human DNA. And of course, in the human DNA, we have an approximately of 25,000 to 40,000 genes. So in genomics, we are studying the DNA and the genes that are found in the DNA. But it depends on the organisms. Here, I'm just actually talking about human genome. But the genome in bacteria is not the same with that of human. The genome in dogs, the human in other species like cow, cats, rats, they have their own genome. And it's because of these differences in genome that is why we are different from them. So this genomics, it allows researchers to understand genetic variations and their impact on health and disease. So we said genetics variations, that is how the genome varies, or how is it different from other organisms? How is it different from other organisms of the same species? Like, for example, human, I'm living in Nigeria, so our own genome might not be exactly the same with the genome of Chinese, from China population, from Japan population, from American population. So we have variation in our genomes, and that is what makes them different from, or that is what actually make us different from them. And also, this genome have impact on our health and disease. Sometimes we actually develop disease due to the problem of our genome. That is why we have genetic disorders. That is the diseases that actually associated with some of the diseases that we have. Like, for example, sickle cell anemia is actually a disease that affects our own DNA. So that is why the genome have impact on our health and disease. And that is why we have a specific type of disease called genetic disease, because it affects our DNA, it affects our genome. So that is genomics, is the study of complete set of DNA within a living organism. So what genomics studies? What genomics studies? So genomics study the structure. It tries to study the three-dimensional structure of the organism's DNA. So if you look at the DNA, it's double-stranded. It is actually a double-stranded molecules. So in genomics, we are actually trying to study the three-dimensional structure of the organism's DNA. And in addition, in genomics, we study the functions of the genes. Like, for example, in humans, we have approximately 25,000 genes. So in genomics, we try to study what are the genes that are associated with our growth, what are the genes that are associated with our development, and what are the genes that are actually associated with function. Like, for example, we have hemoglobin genes, which is actually associated with the functions of the red blood cell. And we have genes like somatotrophin gene. They are actually genes that are associated with our growth and development. So in genomics, we also try to study the functions of the genes that are found in our own DNA. And also, in addition, evolution. That is how genes have evolved over time. Like, for example, the first species of living organisms, according to evolutionary biologists, appears about 400,000 years ago. So how do we evolve? Are we the same with actually the same human that lived like 200,000 years ago? Are we the same? So it's through the understanding of genomics that will give us an idea of how our genes varies from the genes of the same human that lived 10 years, that lived 200,000 years ago. So that is one of the functions of genomics. It will give you an idea of how you can get the DNA of the human species that are living currently and the DNA that actually of those human species that lived 200 years ago. Then you compare them. That is genomics. Then you see how actually the genes change with time, and that is evolution. And then mapping. So in the genomics, it will actually assist, or it will help us in mapping the position of the genes. Like, for example, hemoglobin gene is a gene that actually affected, that actually caused a sickle cell anemia. So in genomics, we can map, we can actually locate the position where exactly that gene is. Like, for example, in humans, we have 46 chromosomes. So mapping in genomics will give you an idea in which of the chromosome and which of the loci. Because even in the chromosomes, we have short arm and the long arm. So it's actually hemoglobin gene is in the short arm or long arm, that is POQ arm. So within the arm, in which loci is it? Because even the loci are numbered. So is it in loci 1 or 2 or 3? So in genomics, it will give you an idea of how you can specifically identify, locate a particular gene. That is the mapping. So, and also editing. Currently now there is what you call editing technology. CRISPR technology is an editing technology that you will identify a particular genes that have any problem, that there is at least some mistakes, like mutation. Like for example, sickle cell anemia is a mutation that actually led to the disease. So with this technology, that is CRISPR technology, you can map the gene, identify the location of the genes where there is the problem, and then you now edit it and replace it with the normal one. And that is the one of also functions of genomics. So then, of course, what are the genomics applications? So genomics applications include, number one, pharmacogenomics. A pharmacogenomics is actually a branch of pharmacy that is associated on how you can actually obtain a genomics information of a particular living organism or of a particular individual to study how that particular individuals respond to drugs. So that is what we call pharmacogenomics. Because I, as human, we might not actually respond to the same drugs, or the response might not actually be the same. So the knowledge of genomics, you can have an idea of one specific individual on how that particular individual responds to a particular drug treatment. That is pharmacogenomics. So you can actually, like me now, you can actually get my gene, or you can actually put me on some drugs. Then after I take the drugs, then you can study some response. That is how I respond to that particular drug through my genomic data. And then we have genome editing. Like as I said, one of the applications of the genomics, you can use gene or genome editing approach such as CRISPR technology to edit a particular genes. Like for example, if you actually, this is some of the technology that you can use it in designer babies technology. You can actually identify a gene that is actually responsible for producing melanin. So you know that the more the melanin content we produce, the darker our skin will be. So you can actually identify the melanin gene, edit it with the CRISPR so that at least you will have the low production of the melanin, and that will lead to the actually development of producing a child with fair skin color. And also at the same time, insect pest control. So the knowledge of genome, you can actually study insect pests such as like Australian sheep blowfly to develop protection against them. Like for example, if you have a plant and you identify that that plant is actually damaged by some specific class of insects, so the knowledge of genome, you can actually edit or you can insert a particular gene in that particular plant or in that particular crop, so that the insects, once they chop or once they come close to that particular crop or plant, they will die. So this is actually some of the applications of genomics. And in addition, what we are going to look at is actually proteomics. So what is proteomics? Proteomics is a large-scale study of proteins, particularly their functions and structures. So in proteomics is an omics approach that trying to study the entire proteins that are found in living organisms, particularly in terms of their function and structure, because proteins functions is related to its structures. So it is a field that is important for understanding how protein works in living organisms and how they relate to disease. So you can extract proteins, you can actually study the structure of the proteins, you can extract it, you can isolate it, and then study the proteins through some of the techniques like immunoprecipitation, size exclusion chromatography, SDS, that is sodium dodecyl Fitch electrophoresis. You can use some of these techniques to study proteins. So ladies and gentlemen, these are some of the techniques that actually we'll be discussing them in Biochem 211 in order to study the functions and the structure of the protein. So these actually techniques, we have immunoprecipitation, size exclusion chromatography. We have also sodium dodecyl. So of course, you can actually decide to digest the proteins, and after you digest the protein, it will give you different peptides. And you can study the peptides through PTM enrichment and peptide fractionation. You can even also decide to separate the peptide using mass spectrophotometer. Then, of course, from there, you can analyze the data to actually understand exactly the amino acid that are present in that protein. So this is actually some of the approach that I use in studying proteomics. So proteomics is the study of the large-scale proteins that are found in living organisms, particularly their functions and actually their structures. So functions and structure are strongly related. So if you have problem with the structure of the proteins, then you will actually have problem with the protein itself. So understanding how the protein works, it will actually give you an insight on how the proteins relate to disease and health. It also helps in understanding the dynamics proteins interactions and their rules in cellular processes. And ladies and gentlemen, let me say this, the functions of each and every cell in our body is due to the metabolites that are found in that particular cell. So that is why understanding biochemistry is very important in understanding the complex nature of a living organism. So if you are able to understand human, or if you are able to understand the metabolite in humans, it means that you are going to actually understand how each and every cells in living organisms work. So what does proteomics study? What do we study in proteomics? So in proteomics, we study the structure of the protein. That is number one. That is how the proteins are folded into three-dimensional structures. Of course, we have four levels of protein structure. We have primary structure, secondary structure, tertiary structure, and quaternary structures. So these are the level of protein structures. So in proteomics, we study the protein structure and also protein function. That is how protein perform tasks like digesting food. Of course, we have enzymes. Enzymes are protein. So how the enzymes digest foods, how the proteins are used in building muscles, and also how the proteins are used in replicating DNA. So in genomics, we study the protein functions, and we also study the proteins interaction. That is how proteins work together inside cells. Because some proteins work in actually in conjunction or in collaborating with other proteins. So that's why we have protein-protein interaction. So in proteomics, we study for protein-protein interaction, and we also study protein expression. So how do we actually study protein expression? Of course, as I said, in human, we have DNA, and in the DNA, we have thousands of genes. So these genes have a potential activity, so it has to be expressed. So how does it express? It's through induction. So you have to use some inducers then to induce the transcription where the DNA or the gene from the DNA will be actually expressed to RNA. And the process in which DNA converted into RNA is called transcription. So the RNA will now also be converted. Of course, the RNA, we have different types of RNA. We have messenger RNA. We have so many of them. So we have messenger RNA, ribosomal RNA, transfer RNA, adapter RNA. We have so many of them. So from the DNA, then it will be converted into, of course, messenger RNA transcript, that is mRNA transcript, through the process called transcription. And then the messenger RNA transcript will be actually converted through the process called translation to produce protein. And this protein have, depending on the type of the protein, if it is enzyme, then of course, will have enzymes activity, which is the phenomics, that is the phenotype here. Then apart from the protein, so this is actually the complete concept of protein expression. So ladies and gentlemen, whenever I ask you protein expression, it means when you convert DNA to RNA, that is transcription, and then from RNA to proteins, which is called translation. So this is actually the protein expression efforts, transcription and then translation. So and then in proteomics, we can also study protein modification. That is how protein are changed, such as through phosphorylation. So sometimes the proteins can change due to addition of some group. It can be methyl group, it can be phosphate, it can be sugar. That is protein modifications. Like for example, in translation modification, some of the proteins that we produce after the expression through the protein expression system, some of the proteins are just unaffected and they cannot do any activity. They cannot carry out any physiological activity until when they are modified. And that is what we call post-translational modification. So now, what are the applications of proteomics? So proteomics have so many applications. Number one, drug discovery. So identifying proteins that could be a target for a new drugs. So in biochemistry, you can actually be involved in drug discovery. You can identify a particular protein that is essential for a particular disease diagnosis. So if you can identify or get a molecule that can block the activity of that protein, it may actually serve as drugs. Like for example, let me just give this example. We have an enzyme that is actually associated with headaches. We call it cyclooxygenase. So the activity or the more the activity of that cyclooxygenase is actually leads to production of prostaglandins, and such prostaglandins, ladies and gentlemen, are found to be associated with headache. So these prostaglandins are produced through cyclooxygenase pathway. So if we can get a molecules that can block the activity of the cyclooxygenase pathway, it means that the prostaglandins will not be generated. And if the prostaglandins cannot be generated, it means that you are not going to have headache. So also at the same time, you can identify a particular parasite. Let's say Plasmodium falciparum, which is a parasite that causes disease in human. So if you are able to identify such types of parasites and identify a protein that is essential for their survival in human host, then you can get a molecules that can block the activity of that. And that is actually the drug discovery. And then we also have disease diagnosis. So identifying some proteins can or that could be used to diagnose or predict disease. So there are some proteins that you can use them that if you see them, it is a sign that there is a disease. So that is why you can use proteins in the disease diagnosis. So there are so many proteins that serve as a marker in the diagnosis of the disease. Like ALT, alanine aminotransferase, is an example of protein that can use in disease diagnosis. And then second, apart from this, then we also have like ALT, alanine aminotransferase, and we also have, we have so many of them. And we can also use it in disease treatment. So developing therapies that target specific proteins involved in disease. Like for example, insulin is a protein, so we can use it in the treatment of diabetes. And we also have crop improvement. So using proteomics, you can actually speed up the development of new crops. So this is actually some of the application of proteomics in omics approach. And of course, look at it. You can actually also study proteomics. There are so many applications of proteomics in disease or infectious disease research. Number one, pathogens identification. So pathogen species and strength identification. With the knowledge of proteomics, you can identify a particular species. So some species of living organisms are identified using proteins. So you can use proteins in identification of species and strength of a particular microorganism. Then also in the same time, intact microbe analysis by native MS. So you can use actually, you can analyze microorganisms with their proteins using mass spectrophotometer, that is MS. And you can also study host protein content. Like for example, let's say there is Plasmodium falciparum infected human. So you can decide to check after the infection of human with the Plasmodium falciparum, what are the protein content that are found in the host. So it is very important. So you can actually so you can decide to say, okay, you can test a particular host, whether it is infected with a particular parasite based on the protein content of the host. And it also has clinical application. So you can use a proteins as a strain-specific biomarkers. So there are some proteins that are specific for identifying, for actually identifying, to identify as a biomarkers. Like for example, let me just give this example of strain-specific biomarkers. Like for example, let's say in cancer, once you have a cancer, you can just identify some proteins. Once you see that proteins, it means that you have cancer. So proteomics is used in that. And then pathogen detection. So with the knowledge of proteomics, you can detect pathogens by PCR or ES/MS, that is mass spectrometry. You can also use it in personalized therapies, because you can actually decide to study my proteomics as human. And then from there, it will give you an idea of how you are going to use that protein's profile of mine for personalized medicine. That is for a specific treatment. So there are some of the disease treatment that you can just only treat a particular person, that my treatment will not be exactly the same that you are going to treat the same another person, even for the same disease. This is because the treatment is based on my profiles. So you can decide to study my proteomics as Mokhtar. Then from that, my proteomics data, then you can actually decide to put me on a particular disease, in a particular drugs or treatments. Then you can also use the proteomics in host cell proteomes. So interaction driving changes by quantitative MS, membrane and cell protein and cell surface protein enrichment, high throughput multiplex MS strategies. So host cell proteome. So you can decide to actually study the host cell proteomes. That is like for example, if you have a particular parasite infecting mosquito, so the mosquito now serves as a host. So you can study the proteomes of the mosquito as a host using quantitative mass spectrophotometer, that is to quantify the proteins. Or you can use multiplex mass spectrophotometer as a strategy in doing that. Then you can also use proteome organization. So proteomics is used in proteomics organization, which is a spatial temporal protein localization. So we can actually use proteome organization to identify a particular protein in a specific location, pathogen remodeling of host membrane compartment, and you can also use it in interaction topology. So you can also use proteomics in host pathogen interaction, like protein-protein interaction or protein-RNA interaction. So this is actually some of the application of proteomics in infectious disease, pathogens identification, clinical application, host cell proteomes, and then proteome organization, which is the last one, is host pathogen interaction. So the next thing that we are going to look at, ladies and gentlemen, is metabolomics. So what is metabolomics? Metabolomics focuses on systematic study of metabolite within biological samples. So when we said metabolites, it can be any molecule that is found in living organism. It can be carbohydrate, proteins, nucleic acid, amino acid, lipid, organic acid, and so many. So studying these metabolites in biological samples is what we call metabolomics. So these molecules encompasses a vast array of chemical compounds, including sugars, including amino acids, lipids, proteins, nucleic acid, organic acids, and nucleotides. So these are the examples of metabolites that are studied in a biological system. So this is actually described as metabolomics. So metabolomics always come first. So after genomics, the next one is transcriptomics, which is from DNA to RNA, that is transcription. So transcription is called transcriptomic or transcriptome. That is the study of the entire RNA, which can be messenger RNA, transfer RNA, and many more. Now, after transcription, of course, is proteome. That is how the RNA will be converted into protein. And then, of course, the environmental influences also with the study of this genome, transcriptomics, and proteomes lead to the generation of metabolites. And these metabolites that are found in a cell will lead to the phenotypic appearance. That is the phenotype, that is the appearance of that particular living organisms. So it means that to study how, or if you want to study how Mukhtar Ali is, in terms of the character, in terms of the appearance, in terms of the behavior, you have to study them from the omics approach. So then the next one is, how is metabolomics performed? So metabolites are analyzed using chemical analysis tools like mass spectrophotometry and nuclear magnetic resonance spectroscopy. So in metabolomics, you actually analyze or you study these metabolites in biological samples using mass spectrophotometry, nuclear magnetic resonance, and so many other. There are so many other techniques. Multi-talk, we have so many of them. So data from metabolomics can be combined with the data from other technologies like genomics and proteomics. So you can actually study metabolomics by integrating metabolomics data with transcriptomics and proteomics technology, so you can study them together. So what are the benefits of metabolomics? So metabolomics can quickly identify and quantify thousands of metabolites. So with metabolomics approach, you can actually study and identify thousands of molecules in a biological sample. And metabolomics can also help to understand the mechanisms that underlie the phenotypes. So with the knowledge of metabolomics, you can actually study the phenotypes of a particular living organisms. So with the metabolites or with the metabolomics data, you can study why am I black? Or why do I have dark skin? Why am I easily developed? Or why am I easily get anger? Why am I intelligent? Why am I short? Why am I tall? So this, with the metabolomics data, you can actually relate this phenotype, my intelligence, my tallness or shortness, my skin color, the size of my nose, and so many others. So metabolomics can also help identify physiological biomarkers that are not approachable with targeted studies. So these are actually these are the benefits of the metabolomics. Then the next one is transcriptomics. So transcriptomics is the study of complete set of RNA transcripts produced by the genome under specific circumstances. So it means that when we say transcriptomics, it means it's the study of complete set of RNA that are found in a cell. So the complete set of RNA, it can be messenger RNA. So when you are studying the entire RNA in a cell or in a particular living organism, that is what you call transcriptomics. So it provides insight into gene expression patterns and regulatory mechanisms. So with the knowledge of transcriptomics, it will give you an idea of the gene expression pattern, whether you are going to have an increase in gene expression or decrease in gene expression. Like adrenaline is a protein. So under some circumstances, you will have high increase in RNA that is associated with adrenaline production. And under some circumstances, it will be low. And that is why you come back to normal after the adenine is degraded, and that means that the expression patterns of the adrenaline gene will go down. And with also that, you can actually study the, you can also study the, with that, you can also study the, you can study, so how the transcriptomic works. Transcriptomic works, how it works, it actually works through one, understanding the DNA. So an organism's DNA contains all the information for the organism. And then the next one is transcription. So studying the transcriptomics approach is always start from the DNA, is because it's after you understand the knowledge from the DNA, or through understanding the DNA molecules, and that will give you an idea of how the pattern of the RNA will actually be. So therefore, in terms of transcription, the DNA is copied into RNA, which contain instruction for making proteins. And then the next one is transcriptomics. So it means that after the DNA will now actually be studied for the entire organism. And then from there, you now study the transcription of that organism through converting the DNA into RNA. And then the RNA contain all the information that is needed to generate proteins. And then the next one is transcriptomics, which is actually the RNA transcripts are analyzed to understand how the genes are turned on and how patterns are produced. So when you said how to understanding the genes can be on and can be off. And when it is on, it can be up-regulated and it can be down-regulated. So that would actually depend on how much of the proteins will produce. So if the transcription, or if the expression of the gene is up-regulated, it means that you are going to produce the proteins for that RNA in higher concentration. And if it is down-regulated, then it is going to be low. So then the next one is, how do we study this transcriptomics? So high throughput methods like transcriptomics uses high throughput methods like microarray analysis and RNA sequencing to study the transcriptome. So we can use microarray, RNA sequencing, or real-time PCR. That is 3 technologies: microarray analysis, RNA technology, and real-time PCR technology to study the transcriptome. That is the entire RNA molecule in a given cell. And you can also do what you call comparative genomics. So in comparative genomics, it's a genomics that you can actually compare the transcriptomes from different conditions to identify genes that are expressed differently. Like, for example, you can take my transcriptome, that is my total RNA before eating and after eating, then you compare it. That is a comparative transcriptomics. So you can take my total RNA before eating and after eating, or you can take my total RNA when I develop a particular disease and after actually the treatment of the disease, you can take the RNA to compare them, and that is the transcriptomic approach. So then the next one that we are going to look at here is epigenomics. So what is epigenomics? Epigenomics examine the epigenetics modification. And when we said epigenetics modification, it's a modification that regulates gene expression without altering the DNA sequence. So you can decide to say, okay, this is the DNA. So using a transcriptomic is an approach that is actually influence the expression of a particular gene, but without affecting the sequence of the DNA at all. What are the sequence of the DNA? That is the base pairs, adenine, guanine, cytosine, and timing. That is without altering them, without affecting them. So that is epigenetics. And then, so this modification can be influenced by environmental factors and lifestyle choices. So the epigenetic modification is depend on the environment. That is why sometimes intelligence, the color of our skin, and some other characters, they are changed with time. Why is it that are changed with time? Because of your life pattern. Like, for example, if at your initial state of from your life history, you started with the difficulty in your life, and then later on, then you have a change in your good lifestyle, you will notice changes. So that are actually some of the examples of epigenetics approach. They are approach that will actually assist and help the DNA to express a particular character, either to increase it, to decrease it, to off it, or to on it. But that's actually due to environmental factors. That's the environment you are in, and also the lifestyle. The lifestyle can be in terms of the types of food that you are eating. So then what are the integrative omics approaches? So an integrative omics approach combines data from various omics disciplines to provide a holistic view of biological systems. So the integrative approach is an approach that can integrate the data from genomics, the data from transcriptomics, from metabolomics, and then epigenetic, and then epigenetics, and then phenomics. You can combine them all together as a single data. Then you can use it to provide a holistic view of the biological system. So this approach enhances the understanding of complex diseases by correlating genomic or genetics, proteomic, and metabolomic data, so metabolic data. So what are the applications of omics in medicine? So omics technologies are transforming personalized medicine by tailoring treatment based on an individual profile. So with this omics approach, you can actually study the profile of one particular individual, and you can actually use that profile to treat a particular individual. So they contribute to the identification of new drugs and biomarkers for early disease detection. So ladies and gentlemen, what are the challenges now that we have in omics? Challenges in omics research: despite its potentials, omics research faces challenges such as data complexity and variability. And standardization of method and data interpretation remain a significant hurdles. So ladies and gentlemen, these are two major challenges in omics approach. Number one, data complexity and variability, because these data that are very complex, and the variability, because the type of data that I have and the type of data that other organisms have, they are not the same. So how this variability in terms of my data and other human data is actually a challenge. And also standardization of the methods and the data interpretation is also a problem. So what are the futures of omics? So the future of omics lie in advancement in technology, enabling more comprehensive and cost-effective analysis. So integration with artificial intelligence and machine learning will enhance data interpretation and biomarker discovery. So one of the actually future of omics data is how artificial intelligence and machine learning will be integrated into omics research, which will actually help in studying or in interpreting the omics data. So these are some of the actually, like, this is food mix. So you see that how the genomics integrated with proteomics, metabolomics, and transcriptomics to study the microorganisms, that is the microbiome, the human health, and some molecules that are involved in triggering so many things. So actually, you see, this is from here, you can say it is a very complicated linkage. So using artificial intelligence and machine learning in data interpretation and biomarker discovery is very important. So ladies and gentlemen, this is the end of this lecture. Please, if this is the first time you're coming to my YouTube channel, try to subscribe and join the YouTube community. Try to join the YouTube community so that you will have access to other videos, lectures. Thank you very much for watching this video, and I hope after watching this video, comment, and that will actually signify that you really watched the video, and we will have further discussion in the class. Thank you very much. Bye bye, and see you later.